All right. So now we have a panel discussion about how to accelerate GBA discoveries, and I would like to call the panelists. So Shalini, you, Angelica, Roy, Simon Stott, and Aviva Rosenberg. So the purpose of this panel is really, we don't have a lot of time. We have 6 people on the panel, means up to 5 minutes each. But we wanted to have a short discussion about how we can accelerate GBA discoveries. So I want to start very quickly, just by each of you introducing yourself very quickly and how what you do is related to collaborative GBA research. We'll start from Kaisa, because it's the easiest. You already said it, I guess, in your presentation, but yeah.
Yeah, so you already heard what I'm working with. I'm a postdoc at Lund University in Sweden, and I work a lot with GB2, and it's, as I said, it is a really highly collaborative effort. It is just fantastic to have colleagues all around the world kind of helping out, with genetic analysis, including an analysis of GBA1, and I highly encourage everyone to kind of have a look at GB2 if you think that any way can, or somehow can contribute to it and be a part of it, because we really want to have you all as colleagues.
I'm Roy Alcalay. I'm the division chief of movement disorders at Tel Aviv Medical Center, and I also hold an appointment at Columbia University. With regards to this panel, my background is clinical and epidemiological. So we collected a lot of samples and we shared them with whoever was willing. Shalini was very helpful with this, and I think a lot of data came out of it from labs that are specialized in it, like glucosylsphingosine in plasma, GCase activity from Sanofi, that did red blood spots. And we're still collecting samples, and if you have a very interesting idea, please hit us up.
Roy, can you say something about PD generation maybe and GBA?
Oh, sure.
Yeah.
Jim will kill me. The other thing is we believe that in order to enable clinical trials, the way to do it is that all people that are interested will know their genotype. If they know their genotype, then it's their choice whether they participate in a clinical trial. And for this, the Parkinson's Foundation launched the PD generation study. And now with the help of ASAP GP2, in 2024, we reached the benchmark of recruiting over 15,000 people and expanding to countries in Central and South America and Israel, ideally expanding further. And you can imagine that the positivity rate for GBA is the highest. It's roughly 8% of the cohort. So these are a lot of people that are going to be very interested in us coming up with GBA-related solutions.
Hi, everybody. I'm Shalini Padmanabhan. I am from the Michael J. Fox Foundation for Parkinson's Research. I'm a neuroscientist by training, but now at the foundation, I head up our discovery and translational research department, so really kind of developing the strategy and implementing that strategy through funding investments in the biology space, in the preclinical development space. So that's all the way until IND, as well as our biofluid biomarker areas. So in terms of collaboration, I would say, of course, a lot of what we do at the organization is to provide funds to academia as well as industry, but by being part of these kind of meetings, we develop a lot of ideas as well. So there's a lot of top-down strategies that we have developed at the foundation and lots of collaborative projects that we have facilitated, a lot in GBA through kind of Roy's center, but many pre-competitive consortia with industry groups that we have done in the LRRK2 space, lots of animal model and tool generation, kind of getting the community aligned on certain assays and biomarker endpoints are all things that we focus on. So I would say we've had a lot of success with these kind of models, more in the LRRK2 space that the foundation has led, and I think definitely coming to this event has given me a lot more ideas. So looking forward to really kind of bringing together a lot more people on more collaborative projects. So I have ideas, but if you guys have ideas, I'm always open to ideas for further advancing GBA research.
Thank you. My name's Simon Stodd. I'm the Director of Research at Cure Parkinson's. We're a small medical research charity focused on disease modification. It's kind of in the name. We were started by 4 gentlemen who had Parkinson's, and they were tired of research, the lack of research not focused on actually curing the condition. And we, within the context of GBA associated Parkinson's, we have been quite influential in the clinical testing and preclinical testing of ambroxol. And we have a phase 3 clinical trial being set up at the moment where we are genotyping, and we're at about 5,000 people at the moment. We're trying to generate a trial-ready cohort for that study and other studies as well, looking at GCase-associated therapies. And we also see ourselves as a convener in the pre-competitive space around endpoints and biomarkers, and having workshops, bringing both industry and academic parties together to have a constructive conversation without the need for keeping secrets.
Hi, I'm Aviva Rosenberg. I'm co-founder and co-president of Gaucher Community Alliance. I'm also a type 1 Gaucher patient and a mother of a son with type 1 Gaucher disease. And so our organization was really founded on collaboration. It was the 3 pillars that make up our group are the patient community, our doctors and researchers, and our industry partners. And so everything we believe, our entire board of directors of 14 members across the US and Canada, and everything we do is founded on collaboration. So I'll just give you a real few examples of some of the things that we do that with. So first of all, we work on clinical trial design from the very earliest stages. So we'll work with our industry partners on sort of unmet needs, what type of, what's reasonable for patients to go through in a clinical trial. We'll work on patient recruitment, and we do that through advisory boards. We convene patients, for specific topics for advisory boards, both for, for clinical trial design, but also for other issues, for social and emotional factors as well, dealing with unmet needs. We also do patient recruitment on samples. So we have collaborative arrangements with both industry partners and academic centers in the US and now GCAN to do patient recruitment for samples. And they could be as varied as GBA carriers or people with NGD. And the final sort of just touch on collaborative that we haven't really been mentioned this conference is we collaborate with the government. And so we've held several FDA listening sessions, and this is particularly important for our NGD community because there's no current treatments for neuronopathic Gaucher disease. And so the listening sessions that we've convened with the FDA is specifically around endpoints. And what does a clinical trial look like if you have variable endpoints, and what's important to the community about a prospective treatment. And so we continuously update and collaborate with our government relations teams on what the needs of the community are. And so we're really hopeful that someone in this room will come up with treatments, and then we can go to the FDA together.
Hi everyone, I'm Angelica Assis. I am from Parkinson Canada, so don't come from too far. And I'm a bit of a smaller fish in comparison to some of my counterparts. But in Canada, genetic testing isn't as maybe compared to other jurisdictions widespread in terms of part of standard of care. So one of the things top of mind for us is really starting to make those first steps in advocating for more genetic screening for patients as we look towards more trial-ready populations for clinical trials. I'm really excited to see all the innovation and all the products in the pipeline, but really thinking about what role we can play to ensure that our population, Canada at least, is ready and mobilized to enroll in these trials as soon as those drugs are being tested in Canada. So one of the things I'm thinking about is how we can collaborate more on this piece around education and awareness, and really particularly educating the patients, the people with Parkinson's, and those who are much earlier now in their disease, RBD, or those who have family members and have now done their own genetic testing, is how do we educate them about what are the opportunities to participate in research, why it's critical to enroll in these studies? What are you contributing? Because I think that's going to be a major hurdle that we need to overcome as a field to really get these populations into studies, because we need large cohorts, as we've heard repeatedly today, to really power these studies to look at those correlations, to test drugs. So I think that's a role that we as charities can play, is really that education and helping to provide the supports where needed to ensure these populations are ready to enroll in studies.
Thank you. So my first question to the panel is, what do you identify as the major gaps in GBA research that can be answered by more collaborative research?
Could I interrupt for just a second and ask a maybe simpler question and then go back to that one? Because this is going to be such a large operation with enormous amounts of medical information, and having just experienced Morey Hospital in Chicago being shut down for 2 weeks because of a hack, what is the security around the data that is going to be in here and the identification of patients? You probably don't have a plan right now, but what would you be doing to make that plan?
I'm sorry, Greg, but I have to stop this because the topic really here is not that. I mean, data security is very important, but I really don't want to go into this now. I'm sorry.
It's a question of whether
Yeah.
you're doing anything in that regard right now. You don't have to lay it out for it, but that would be a real hesitation.
I understand, but that's not the purpose of the panel right now. I'm sorry. Go ahead.
Okay, so well, actually, to Greg's questions, we don't collect any data. A lot of the patient advocacy organizations, we do not collect data, and so we answer it by that, is that we rely on, when we recruit patients, we rely on the studies or the recruitment, the academic centers or the industry partners who's then collecting data to have those types of security systems, which are absolutely necessary, but definitely out of the price points for patient advocacy organizations. In terms of collaboration, the way that where we would like more collaboration, and we've seen the last 2 days have been amazing in sort of the neurodegenerative space, is that we would love to see some of that collaboration trickle down to the neuronopathic Gaucher disease research, because if you have more severe mutations are leading to earlier Parkinson's, and if there's something available for that to slow that down, can we take some of that research and can we also test it out on the neuronopathic Gaucher community around the world, which currently has nothing? So that's where I would like to see more collaboration.
So with regards to the gaps in research and leading to opportunities for collaboration, one of the obvious, it's quite a stark fact, is that in this room we've only got 2 patients representatives for the Parkinson's associated GBA cohort. One of them is a fantastic advocate for research in this field, and the other is Ben Stetcher. But at Cure Parkinson's, we're always about the patient voice, and you have to have that representation in the room. Even if you're talking complex molecular biology, there has to be that reason for doing it. And I think there's a massive opportunity to engage with the patient voice, and from that, there'll be opportunity for collaboration as well.
So for me, I think Ziv, we covered it really well at the SAB meeting. I think there were many points that were raised by many speakers over the last 2 days, and I agree with maybe Roy. First, we really need to harmonize where possible. I think harmonization of cohorts, harmonization of our preclinical resources, just making it very convenient for the community to access these resources and develop these collaborative initiatives would be key, I think, in my opinion, and that's quite a low-hanging fruit with initiatives like GP2 and PD generation and things like that. So definitely that's one area I would see ample collaboration potential for. And then the second one is, I mean, I was telling many people, I just came back from the LURC2 meeting last week, and we had 4 companies at the LURC2 meeting, and there's 26 companies here, which is amazing. So it's really nice to see so many industry groups here, interacting so closely with the academic community. And we have felt in the past that our collaborative efforts that included clinicians, included industry groups, as well as academic groups have been the most successful. So definitely I would anticipate kind of, having more collaborative efforts around our biomarker initiatives, focusing on GBA through these different stakeholders. So that, I think, is what I see.
So I think one of the major gaps working together can help is really the more we disseminate the knowledge about the genetics so people know their genotype, if they're interested. If people don't want to know, I think it should not be coercive. But we find out, just like the court from Nigeria said, and it's true with every court, that people are actually more interested than we can imagine. The more people know their genotype, the more they're willing to participate in studies, biomarker studies, clinical trials. The more clinicians know patients, the more observations they can make. DBS works or doesn't work. So knowledge leads to knowledge, and that would be one place where I think we should continue those efforts of genotyping. And the second point that I will briefly mention, because Shalini addressed it, is that I think working with pharma, there's wealth of knowledge in pharma that we should just disseminate. If people know that their assay is working, to work with academia and publish it, and then so efforts are not replicated, or if things don't work, to know that they don't work. So these would be the 2 key points.
Yeah, not sure if I have so much to add, but I do agree that I think it's very important with the harmonized data sets. And I do think, for example, then if GP2 can kind of help out bringing in the genetic data and make sure that, we're using the same array for all of samples, and kind of course, then when we have the results, when we have, when we have checked accuracy of the array as well for the GBA1 calls, I think it could be really important to bring that in and also kind of function as a platform even more for collaborations.
Kaisa, do you want to say a word how in GP2 data is being secured?
Yes, I will try, to my knowledge.
And you can chip in too if you want, or Cornelius.
Yeah, so we're working with Google on this to have secure platforms, and all users have to sign data user agreements, and we have to have 2-factor authentication to log into the servers. We also have GDPR compliant servers in Europe, but Andy, please fill in further.
Yeah, I think you covered it. So we use a platform that is part of the Accelerating Medicines Partnership Program, which is a US government and pharma and foundation initiative. So as you can imagine, a huge amount of work has gone into securing data. So yeah, I think that's a space we've worked on really well. Something I hinted at earlier is that if you're going to do a global collaboration, there are data types that can't move out of certain domains. So we've also worked at creating an infrastructure that allows data to stay in Asia or Europe or the US or South America to ensure that we don't cross any data boundaries.
So my next question is to Shalini. I would like you to start, because you brought this up, the harmonization of data. How will you go about it? How can it be done in an efficient way, because it will be a lot of work, right? And many centers should be involved. Not harmonization of data, sorry, making sure that the different assays are being done the same way in different places, and standardizing protocols and things like that. How should we go about this?
Yeah, I got worried when you asked me about data. I was like, okay, I'm not the data person. So for assays, actually, a while ago, we realized that we had quite a few assays for GCase, right? I mean, used in academia. So I think the first step, I would say, always is to bring, certain KOLs together, kind of align on what the problem is, and define what the best solution would be, and then kind of work backwards on the approach. So I think Dan from Vanqua helped us kind of lead some of these efforts early on when we said, let's kind of just evaluate the landscape and see what are the GCase assays out there. And that kind of got a team of investigators and KOLs from the field together to define what those assays were. And then we realized, okay, we really need to kind of move forward and kind of develop better assays. So for us at the Fox Foundation, we always kind of say, you know, what is the problem? What would be the best solution and how do we get there? And then kind of when we realize, how, you know, the multiple steps that it takes, we kind of figure out how we can shorten that that timeline by kind of bringing collaborators together. So just to give you an example, I think we were looking, we did this whole kind of evaluation of the lipid assays that are out there in the community, and there were 3 or 4 companies that came to us with their assays, their sphingolipid assays. And then we said, okay, the best test would really be looking at GBA carriers, and then also have like throw in some Gaucher carriers there, so that if we can actually distinguish, you know, PD from GD, it would be kind of a good sign. And so with Roy's, so that's how we found Roy, and Roy already had all of these samples. We sent that all blinded, and we unblinded them, and then we could really clearly see which assay kind of performed better than the other. So I think it's these small kind of studies that kind of give us confidence on how we can then go bigger. So now we already know 2 or 3 companies have like good assays that can eventually move into cohorts like PPMI. So for us, it's always defining the problem and defining what the best, what success looks like, and then figuring out the approach in between that usually involves all stakeholders. So it becomes big. It's a consortium style approach. Hopefully that's helped.
Yeah,
I don't know
thank you.
if you wanted me to.
Anyone else from the panel want to answer? Okay, so
I will just say that it's the pharma interest to help these efforts, so then when clinical trials are developed, they don't need to reinvent the wheel about their CRO and like there is a consensus in the field that this is a good system to measure GKs, or a good system to measure glucosylsphingosine, and that it's something that is neutral. So it's a win-win if you get people to do it together.
So one major gap that exists, and it came up many times already in this conference, and for example, the best example may be Lara's talk about the African variant. She mentioned that they don't have really polysomnography ability, they don't have genetic screening is difficult, they don't have genetic counseling. So what can we do in order to make things better in places that, how can we help? What should be the approach? So we know GP2, for example, you're already kind of doing this by trying to prioritize underrepresented populations, but how else should we do it more on the clinical aspects, and such as genetic counseling and you know, clinics, RBD diagnosis, etc.
I mean, I'm not going to say too much if it's wrong, but GP2 is also now, there are free online courses that you can take in GP2. A lot of them are focusing on how to analyze genetic data. But I do think as well that there are some shorter courses coming out that might be beneficial for the clinics as well. Kind of how, maybe not how to diagnose, but how to think of some, for example, a typical Parkinson, the different diagnoses. So kind of also to harmonize how that has been done all around the world. Yeah.
I will let others from the panel to answer, and then I will let you ask it.
I think one of the challenges with, you know, aside from doing international type of research, is the recognition that once you have a diagnosis, there may not be the treatment availability for that patient. And so, you know, it's obviously you want to have a diverse as cohort as possible, but you have to remember that the family that you're involving, they get this diagnosis, and then what? You know, in some countries around the world, you have individuals dying of type 1 Gaucher disease. And so coming from the Western world, that's crazy, right? There's been treatment on the market for almost 30 years. So it's just sort of to be really careful, like, yes, research is cool, and like these different mutations from different places are really cool, but what does that actually mean for the patient families then, you know, when they hear about this? And if there's not a next step for that, you know, it's just a question of what, you know, how to bridge those two gaps.
Anyone else from the panel wants to?
I mean, I'll say recently we started to work very closely with our fellow charitable organizations, Michael J. Fox Foundation, groups as well from the Alzheimer's and MSA space to form a coalition. So this is still in its infancy, but I think these are the opportunities where we can work more collaboratively, come together to exchange ideas, talk about what's going on in our respective jurisdictions and countries. But I think this could be an opportunity to also look globally at what are some of the infrastructural differences, what are some of the other differences in just societies, in cultures, where we could start looking to pool resources and set priorities in terms of funding in specific areas or funding for specific training opportunities to start ensuring that there is more of this global engagement and research. So I think something like a coalition where we're working together, pooling resources, looking at a global view, there could be opportunity to address some of these challenges of inequity or differences in practices.
So in January of this year, I was invited and spoke at a conference, a really large genetics conference in Ahmedabad in Gujarat in India. And I learned, which I didn't know before, that there's probably an estimated 25,000 patients, potential patients with neuronopathic, to speak to Aviva's point, with neuronopathic Gaucher. And the conference this year was hosted by the Fridge House, which is it's an amazing place run by a family, and they are the foremost genetic diagnostic centers in all of India, and they have a huge amount of data, huge amount, and growing all the time. And I was thinking that, to Aviva's point again, that they should be part of this collaboration because, as she said, there are so many people who already are diagnosed with both type 1, and I've seen these people, type 1 and type 3, that have a diagnosis but are not able to access treatment. And it's a horror, it's a terrible, it was very painful for me to see this. And so I'm really hoping that you all reach out to them and have them join this wonderful group.
Ellen?
Yeah, it's sort of an uncomfortable subject, and it's way off. But part of what I think as a community is going to be important is to try to do something to ensure that when there are therapies, they're accessible and affordable. And that's sort of been the problem of the Gaucher community. And there's nothing we've ever been able to do about it. But perhaps if this is something that's considered up front, it would be helpful.
Any comments from the panel?
I think you're absolutely right. It's like there is always this challenge because it's also like very practically, I think, for pharma, the question is, are they defining the treatment for Gaucher, for PD, or for GBA-PD that would be designated an orphan drug? So that's and then the prices can be very different. It's just like your comment is very optimistic because you're talking about the future drugs that are not there yet. And so I really hope that this becomes an issue. Right, but I totally agree. I wanted just to mention another comment and see what the people in the panel and the audience think about it. I think it's on one hand very important to harmonize assays and to make sure that we measure GKs correctly in the lipids, but it's also very important to keep the field open for different ideas, so we're not dogmatic, thinking like in one way, and then anyone who is not thinking in the same paradigm is kind of their voice isn't heard. So, because we need to understand that we don't really know a lot of things about GBA-PD, and we need to have also people thinking outside the box to solve this question.
So I completely agree with that, Roy. And in fact, I think ASAP the Aligning Science Across Parkinson's was launched 2018. And I think really the goal, and they have, of course, many arms. They fund the Global Parkinson's Genetic Study. They also kind of support PPMI. But one of the arms that ASAP supports is the Collaborative Research Network, which is really this huge network which is focused on team science, multidisciplinary science, focused on the mechanistic basis of Parkinson's disease. And I think really the goal there was to give people kind of the money and huge amounts of investments, and let them really kind of come up with bold ideas and risky ideas and take that through the 5-year period, the 3 to 5 year funding period. So I think those kind of initiatives are emerging, and just wanted to let this group know that later this year in 2024, you will see kind of new themes for the next round of ASAP launch. So if y'all have out-of-the-box ideas and y'all want to kind of really kind of, put together a team of investigators to answer these kind of questions, I think ASAP would be kind of the best place to go to. But there are people kind of coming up with that. So I think that's we always have to do both in parallel. I agree with you. I don't know if anybody else wants to comment.
All right, thank you very much. So with this, we will end the session and end the day. So thank you very much to the panel members and to all of you, and see you tomorrow. Huh?